Polytechnic School in Zurich, which is a very famous interdisciplinary school, research institute in Switzerland. And then he had his Ph.D. at Weizmann Institute in a very famous group with Dr. Elinav. Then I believe went for a short time to Harvard, and then has been recruited as an assistant professor at UPenn. And his lab is very active, and this is what he's going to explain, on investigating the impact on the gut microbiota, the intestinal microbiota on host physiology, and on the interplay between the host and the environment and the impact on a metabolic disease. I should also tell that his wife, Dr. Mayan Levy, is also a very highly productive young investigator in immunology. And we were planning to have both of them coming at USF, but COVID-19 has dictated against. But Mayan will give a seminar on August 20 in the context of the hub on microbiome, and we also very much look forward to this seminar. So Christoph, thank you again for being with us today, and we welcome your lecture.
Thanks very much for the kind invitation, and I'm really happy it worked out now despite the pandemic, even though we didn't get a chance to visit in person, but hopefully we'll be able to do this as soon as the crisis is finished. Let me share my screen. To start the presentation. So if you can see my slides, what I want to highlight right away in the beginning is that there are really two major themes that we're studying in my lab. The first one is, as you can imagine from the introduction, we're very interested in host microbiome interactions. We're studying these from multiple different angles with a focus on multiple human diseases and how the microbiome contributes to their pathophysiology. But at the same time, and of course this is now becoming really relevant in the context of the current pandemic, we're interested in a phenomenon called disease tolerance, which is basically teaching us ways to approach infectious disease and other diseases in a non-traditional way, and trying to see if we can boost disease tolerance and thereby enhance survival of the host to different microbial insults or to different other diseases. And I'll talk a little bit about this in the second part of the talk. But for now, let me focus on host microbiome interactions. And if I can go to the next slide. Okay, here you will see this is the major area of interest in my lab, which is now again very relevant for the current pandemic, because we see that this infectious pandemic is meeting the metabolic pandemic of obesity and diabetes. And as we have learned very early on in the current pandemic, obesity and diabetes are among the main risk factors for severe cause of COVID-19. What you can take away from these slides, which I'm sure many of you have seen already in multiple shapes and forms, is that this is really a global situation that has been going on for the last few decades. We know that it's happening in men and in women. We know that it's happening in basically every part of the world. And we know also that it happens in every part of society. So this is really something that we haven't found any good scientific or medical solution for yet. And you can see that the numbers are rising almost unstoppably. The other important takeaway from this graph is that this has been, in terms of the history of humankind, it's been a very recent development. You can see that basically this explosion has happened really over the past 3 decades or 4 decades. And for us, the main conclusion is that this is likely not explained by a change in the human genome, because the human generation time is just way too long to account for this massive susceptibility in the human population. And instead, we usually conclude that it's primarily environmental factors that are influencing our susceptibility for this massive rise in metabolic disease, including the ones listed here, such as nutrition, changes in circadian rhythms, control of temperature, lack of exercise, elevated levels of stress, and exposure to xenobiotics. Because I'm sure all of you will agree with me that these 6 environmental factors and some others have strongly changed in their composition and the extremity of their exposure over the last few decades, in which we have seen this massive rise in obesity and diabetes. What's interesting to us from a scientific standpoint is that there's actually very little scientific knowledge about how these environmental factors influence the human organism in a way that so strongly predisposes us to these metabolic diseases. And that's something that we're really interested in my lab. And what we're working on specifically, as Christian mentioned in the introduction, is the role of the microbiome in mediating some of these host susceptibilities to environmental changes and environmental factors. So we and others, many other groups in the field have worked out over the last 5 to 10 years that there is a very strong interaction between the microbiome, environmental factors such as diet, and host factors such as the innate immune system in mediating some of these susceptibilities to disease. And you can see here that this is not only influencing metabolic disease, but goes as far as other inflammatory and even neoplastic diseases. The technologies we use to address these questions in the lab are sequencing-based technologies, including sequencing of DNA and RNA of the microbial community, as well as HPLC and mass spec-based technologies for interrogating the proteome and metabolome of the microbiome. And then we're coupling this to functional readouts, which we can do in anaerobic chambers or using imaging of the microbiome in mice and humans. And more specifically, what we're doing, of course, is using germ-free mice, which allow us to do functional experiments and to very specifically interrogate the causative function of the microbiome in multiple disease contexts and in mediating environmental influences on host susceptibility for disease. So with this toolbox in hand, what we can now do is we can come back to this original question of how different environmental factors predispose us to developing some of these very common human diseases like obesity and diabetes. And what I'll do in the first part of the talk is I'll share one specific story that we've been working on how nutrition influences the microbiome and thereby influences our susceptibility for metabolic disease. Because I'm sure all of you will agree with me that we have a very good agreement by now that nutrition is probably the #1 factor shaping not only our susceptibility to metabolic diseases, but also shaping the community of the microbiome and its composition and function in multiple ways. And of course, this has been extremely dramatically been influenced by the change in human nutrition over the last few decades. So when we talk about obesity and diabetes, usually what we do, what most labs in the field do is comparing these situations. So we have, let's say in a human cohort study, we have a cohort of lean controls and we have a cohort of morbidly obese individuals. But this is likely not reflecting the reality of human obesity, because the reality of human obesity looks something like this, where we have many intermediate states of these extremes. We have people gaining weight and going on a diet, losing weight, then bouncing back to their original weight, and in some cases even higher. So this is the more accurate reflection of obesity and diabetes in the human population, where we have a gradient of states anywhere in between a low BMI and a very high BMI that is associated with metabolic syndrome. And if you want more scientific evidence than my cartoon here, I would refer you to a very popular TV show in the US, which is called The Biggest Loser, which has run for a decade or so, I think, and has really basically awarded cash to people that lost the biggest amount of weight during the TV series. So there were people that started out like this, and at the end of the show, they looked like this. And if they lost more weight than anybody else, then they got a cash award. And the scientific analysis in this case was actually done by the New York Times, because what they have done is they have followed the weight of these participants. This is one particular season of this show. And you see that during the show, they all lost weight. But then in the follow-up, 6-year follow-up, you can see that almost all of them regained weight, in some cases even higher than the obese weight they had started out with. And you can see that this is a general trend of not being able to maintain this low weight. There is one exception here, which is a participant that cheated a little bit because it was a case of bariatric surgery. But in almost all other cases, they have trended back to their original weight. And of course, this is phenomenon that we know really well in the human population and in obesity research, which we typically call the yo-yo effect of recurrent obesity, which basically describes this failure of obese individuals to successfully maintain a low weight after a diet, and instead they bounce back to the original weight. So a number of years ago, when we started this project, when I was a graduate student in Aran Elinav's lab in Israel, we thought that it was actually surprising that there was so little scientific knowledge about this phenomenon, about this very strong propensity of humans to regain their original weight after a successful diet. So to address this, we designed a very simple mouse model, which allows us to study this in the context of the lab, where we have, first of all, we have two controls. We have this black lean control here, and then we have a high-fat diet group that gains weight over time and becomes obese. And then we have the interesting group here, which is the one that undergoes these cycles of weight gain and weight loss and weight regain, which is reflective of the human situation in most cases. And we have another control, which is the green one here, which is exposed to high-fat diet for the first time only here, when the red group is exposed to high-fat diet for the second time. So we basically have a situation where we compare a first-time obese group to a second-time obese group. And you can see that very similar to the human situation, a history of obesity in the red group leads to an accelerated secondary weight gain here, which means that basically we have a similar phenomenon here in mice as in humans, where we have more accentuated cycles of weight gain every time the high-fat diet comes back. And we can do this for multiple cycles. We can continue this not only for a second, but for a third cycle. And you can see that with each additional cycle, the response is more dramatic. And you can see that every time the high-fat diet comes back, there is accelerated and more accentuated weight gain. Now, this is, if you show a graph like this to an immunologist or to a microbiologist, they will tell you that this looks exactly like a memory response, especially people studying CD8 T cells and now in the context of antiviral responses, this has become very interesting again to see that basically in the context of T cells, we have here an expansion phase, then a contraction phase, and then there is a memory phase. So anybody studying immunology would call this a memory phenomenon. So even though here this is a metabolic disease phenotype, we wondered whether there is a similar concept here of a memory being stored at this time point in the red group, which basically recalls this response to a secondary metabolic challenge that comes here, which then provokes a stronger response the second time around. So for a long time, we were looking everywhere for this metabolic memory. We went through all the parameters of metabolic syndrome, obesity, glucose tolerance, insulin resistance, sedentary lifestyle, food consumption, and many others. But to make a long story short, we haven't found this metabolic memory anywhere in these parameters. You can see that the red group at this time point, after successful diet and after successful weight loss all the way down to the black control, is indistinguishable metabolically from the black control. We weren't able to find in any of these parameters a metabolic memory. The only place where we did find a memory, though, was in the composition of the microbiome. And this is what you can see here. So here we're basically looking at a PCA analysis over time. So each dot represents the microbiome of an individual mouse. Before obesity, they all cluster together. During obesity, they cluster separately, as we would have expected from the work of Jeff Gordon and many others. Obesity is associated with a distinct microbiome profile here in these high-fat diet fat mice. But now, interestingly, at the time point where I just told you we cannot distinguish between the red group that underwent a successful diet and the black group that was lean throughout, we do have a distinction in the microbiome. So the red microbiome ends up in this intermediate space here. Does no longer look like the high-fat diet microbiome, but also doesn't fully recover to the lean control. And this is also reflected by alpha diversity read out here with the CHA1 index. You can see that, as we would expect during obesity, these levels plummet. Diversity of the microbiome goes down, as we would have expected from previous studies. But then interestingly, after obesity, when the red group, again, is metabolically fully normal, the microbiome does not recover and actually stays in this poor diversity state here. So after seeing this, of course, the question was, is this functionally involved in this second cycle phenotype? Is this the reason why there is this strong susceptibility for accelerated weight regain? So we did a few experiments to address this. The first one is what you can see here. So here we're asking how long does this memory, quote unquote, of the microbiome last? So here we have another experiment where we have this initial cycle of obesity. And then instead of coming back with a high-fat diet, we just wait and wait and wait and sequence the microbiome along this time course here to see how long does it take the microbiome to normalize. And when we do this analysis, and here we're looking at distance, basically taxonomical distance between the microbiome of the red group and the black group, which is the baseline here, then you can see that it takes 21 weeks, a very long time after this initial cycle of obesity, for the red microbiome to become indistinguishable from the control microbiome. While of course the microbiome of the blue group, which is this obese group here, stays distinct throughout. So it takes a very long time for the microbiome to normalize, but what's now even more interesting is if we wait for the microbiome to normalize, and then we come back with a high-fat diet, we no longer see this second cycle phenotype. So if we wait enough for the microbiome difference to go away, the secondary susceptibility also goes away. And now if we come with a high-fat diet, we don't see any difference anymore between a primary weight gain and secondary weight gain. We can address the role of the microbiome in different ways. One would be, so here we just waited for the difference to go away. Another option would be to use the sledgehammer approach and to kill any differences with broad spectrum antibiotics. So that's what we did in this experiment. We wait, we don't wait in this case, but we kill the microbiome difference that arises during this period by broad spectrum antibiotics. And then we come back with a high-fat diet. And again, there is no difference anymore in secondary weight gain if we normalize the microbiome here with antibiotics. And then finally, basically the gain of function equivalent to this loss of function experiment here would be to transfer the microbiome to a new recipient. And this is what you can see here. So now we're adding a fifth group to this experiment, which is this orange group here, which first of all behaves just like the red group initially. It has this cycle of weight gain and weight loss from this high-fat diet feeding cycle. But then instead of the red group, what we did here is we transplanted into this yellow group by fecal microbiome transplantation, the microbiome of a healthy mouse. So now this yellow group or orange group here receives non-obese microbiome. And now you can see it behaves like the green group. So it forgets that it had this previous history of obesity, and it gains less weight than the red group, which receives a controlled fecal microbiome transplantation from itself, basically. So with all these different strategies, we were able to prove that the microbiome is involved in this second cycle phenotype of exacerbated weight gain after a history of obesity. But if there's so much information in the microbiome at this time point here, then we thought maybe we can do something more informative, which is to use this for prediction of the outcome. So we teamed up with a group of Eran Segal, who is a mathematician, and especially Daphne Rothschild in his lab, who generated a machine learning-based predictive algorithm, which is taking the 16S information at this time point. So we did another of these experiments that you know really well by now. We basically have this cycle of weight gain and weight loss. You also know that the microbiome is distinct between these two groups. So the ex-obese mice have a different microbiome compared to the controls. And then we're using this algorithm to not only predict which mouse was obese in the past, because I told you metabolically, it's very hard to distinguish between these two groups because there is really full recovery of weight and other metabolic parameters at this time point. And we do something which is even more challenging, which is to predict the exact extent of future weight gain when the high-fat diet comes back, only based on 16S information. So you can see the results here. We can almost with 100% accuracy, we can predict, again, only based on taxonomic composition of the microbiome, which mouse had a history of obesity. So we can basically, by looking at the microbiome, we can tell the previously obese mice from the controls. And with 72% accuracy, we can predict the extent of weight regain when the high-fat diet comes back. So again, just based on the information of the microbiome, we can predict how much weight gain will take place in the future. And then if there is so much information, not only for prediction, then there must be a mechanism that can explain this, this microbiome effect on the susceptibility to a second weight gain. So to understand this mechanism, we focused on the factor of the microbiome, which is probably the strongest in terms of influencing host physiology, which is the entirety of small molecule metabolites that the microbiome produces, and that gets access to the metabolic system of the host. So we performed untargeted metabolomics in over a time course of weight gain and weight loss, and then we classified each metabolite according to its temporal pattern. So you can see here that we have metabolites that are reversibly lost, so they go down during obesity, but upon metabolic recovery during this quote-unquote memory phase, they come back to their original state. We have the opposite. We have the ones that are gained during obesity, but they are fully recovered afterwards. And then we have these two interesting groups up here, namely the ones that are gained during obesity and they stay high, and the ones that are lost during obesity and they are never recovered. So these are basically irreversibly changed metabolites, and we can look at them in a different way by plotting them out against these two axes. And what we see now is that there are two really interesting candidates here in the group that is persistently lost. So this is this first group up here, which are lost during obesity and they never come back. And the reason why they're interesting is because these two molecules, apigenin and arginine, first of all, they both belong to the same chemical family of molecules. Both of them are flavonoid molecules. And second of all, these are molecules, polyphenolic molecules that have been associated in numerous epidemiological studies with the ability to maintain a low weight. So we thought maybe there is a causative link here by which we can explain this susceptibility for the exacerbated weight regain after a history of obesity. So that to directly test the role of these two molecules, we performed another of these experiments where we supplement these two molecules. So again, oh, it seems like now my slides are stuck for some reason. Okay, there you go. So here there is, this is the experiment I wanted to show you. So here we have, again, this yellow group, which as opposed to the previous experiment where we provided these mice with a healthy microbiome, now all we do is we supplement these two molecules, apigenin and arginine. And we are trying to see what's the effect of this supplementation of the lost flavonoid molecules to the weight gain phenotype. And what you can see here is that, again, the yellow group first behaves just like the red group. So it gains weight normally, it loses weight normally. And then when we start the supplementation of apigenin and arginine, now again, they behave like the green group, which sees the high-fat diet for the first time. So just by supplementing these two molecules, we can make them forget this initial phase of obesity and behave like a mouse that sees the high-fat diet for the first time, and basically ameliorate this accelerated weight regain phenotype here. And then finally, one more piece of data that I want to show you before I move on to the second part is that we have initial insights into how these two molecules might mediate this phenotype. So we looked at multiple metabolic parameters that could explain this difference in weight gain here. And we ruled out differences in food intake. We ruled out differences in most aspects of energy metabolism. But there was one parameter which was consistently different, and this was energy expenditure. So you can see here that as expected, the high-fat diet mice have reduced energy expenditure because they're obese. And what's interesting here is that the red group, at the time where they are metabolically completely normal, after they completed the successful weight loss period, they have reduced energy expenditure as compared to the two controls, the ones that stay lean throughout and the green one that sees the high-fat diet for the first time. Now, what's interesting here is that all the interventions that I've shown you are successful in treating this condition, including antibiotics as well as supplementation of apigenin and arginine down here. They basically restore energy expenditure. So now this, in both cases, the yellow group behaves just like the green and the black group that have been the two controls. So we can restore energy expenditure by either antibiotic treatment or by supplementing apigenin and arginine. And when we talk about energy expenditure in the context of weight gain, immediately what we think about is brown adipose tissue. And within brown adipose tissue, a factor called uncoupling protein 1 or UCP1, which is in the mitochondrial membrane, basically bypassing the gradient of protons that drives ATP generation, but doesn't, instead of generating ATP, it generates heat and thereby contributes to thermogenesis. And you can see that in both interventions, again, with antibiotic treatment and with apigenin and naringenin, we boost the expression of UCP1, both on the transcriptional level as well as the protein level. So we think that this might be a potential mechanism by which either antibiotic depletion or supplementation of apigenin and naringenin might have this very strong effect on thermogenesis and preventing weight regain. So just to summarize what I just showed you, we have basically this memory effect in the microbiome after successful weight loss during this memory state. What the microbiome is doing is degrading these two flavonoid molecules, apigenin and naringenin, and thereby contributing to decreased energy expenditure, which is then driving accelerated weight regain when the high-fat diet comes back. And we can use this information for the computer-based prediction of weight regain, but we can also use this information now to start thinking about potential treatments for this condition. I've shown you experimentally this works really well by fecal microbiome transplantation or by supplementation of these two molecules, apigenin and naringenin, and potentially other flavonoid molecules. So with this, I want to switch gear here a little bit and go to the second part of the talk, which will focus on disease tolerance. And I'm highlighting this here, of course, in light of the current pandemic and trying to see whether concepts that are coming from this relatively new field of disease tolerance studies can be informative in the case of SARS-CoV-2. So traditionally, we think about, as microbiologists, we think about microbial load being associated with disease in sort of a one-dimensional space, where higher microbial load is associated with disease and the reduction of microbial load leads back to health. And of course, you know, a century of microbiome, of host microbial interaction research is based on this paradigm. All of Koch's postulates are based on this paradigm, where we have basically identification of the qualitative microbe in a diseased state. The absence of the microbe will be associated with health, and we can transfer the disease by microbial transfer. But I think what we're realizing in many different contexts, and of course especially in the current pandemic, is that this one-dimensional state here is a dramatic oversimplification, and the reality of microbe-caused disease is a lot more represented by this scheme here. So where we actually have a two-dimensional interaction between pathogen load and health. Because what we see currently is that in many cases that are asymptomatic after SARS-CoV-2 infection, we have very high pathogen loads, but they're still associated with health. But then there is a second axis, which is the health axis, which brings us down to this disease area, even though the pathogen load is not necessarily changed. And then again, there is the recovery trajectory here, which is initiated after pathogen clearance, but then again, basically at the same low pathogen load, we need to do something along the health axis in bringing us back to a good state of health, as opposed to staying down here in the low health or diseased area. This is a concept coming from David Schneider's lab at Stanford and many others that have followed up on this. But I think in the current context of the current pandemic, I think it's extremely relevant to start identifying what brings us up and down this health axis here, as opposed to simply what prevents infection or what can sterilize the virus once it has entered the body. Because I think the first few months of the pandemic have taught us that it is probably insufficient just to target this axis here, which of course is extremely important by itself in terms of finding antivirals and finding ways to enhance immune clearance of the virus. But at the same time, we need to strongly work on this axis here and understand first of all, what distinguishes between healthy cases and disease cases of the same pathogen load, and also what brings us back along this recovery arrow here. And just to emphasize this point, this was a paper in Nature Medicine relatively early during the pandemic, where you can see that there is actually no distinction between mild and severe cases of the disease after disease onset. So here they're looking at the levels of SARS-CoV-2 in nasopharyngeal swabs by qPCR. And you can see that the red line of severe cases and the blue line of mild cases, they're basically indistinguishable as they clear the virus and continue this trajectory after symptom onset. So based on, this is again another argument for saying that based on pathogen load itself, it wouldn't have been able to distinguish mild and severe cases in this specific cohort of patients. But there must be other factors that are distinguishing here. And of course we know, and I already told you in the beginning, that one of the major factors distinguishing between mild and severe causes is obesity and diabetes as major metabolic risk factors for the disease. And this actually goes back to another study that I want to share with you that we have been working on a while ago to distinguish the role of metabolic disease in, or to decipher the mechanisms by which metabolic disease plays a very important role in regulating our susceptibility to infection. So this goes back to this study, which you can see here, and many others of the same kind that have documented positive correlation of BMI and susceptibility to infection. So here we're looking at overall outcome of infection in men and women stratified by BMI of the host. And you can see that the BMI higher than 30 is associated with a higher hazard ratio for these infections. And again, very similar to the first story that I told you about, we decided to model this process in mice. So we get a very mechanistic angle at understanding what happens there. And we used a very standard obesity model, which I'm sure many of you have seen in the past. This is the DBDB mouse. It becomes massively obese, as you can see here. And the reason why it becomes massively obese is because it has a mutation in the leptin receptor. So these mice don't feel satiety as their litter mates, and they basically don't stop eating. And what's interesting here is that these mice are extremely susceptible to infection. So here we're infecting them with a bioluminescent version of Citrobacter rodentium, which is a GI pathogen. And you can see that while a wild-type mouse is able to contain this in the GI tract, in the DBDB mouse, we get massive spread to the mesenteric lymph node, the spleen, and the liver. And it's interesting to point out that the colonic levels of the actual infection are unchanged between both conditions, which means that we have a typical example here where pathogen clearance is actually not affected at the primary site of infection, but disease tolerance is affected in terms of locally containing this pathogen and preventing systemic spread in the context of metabolic disease. You can see here that this is associated with loss of intestinal barrier function because these DBDB mice, they have lower levels of many of these components of the intestinal barrier. You can see this here also quantified by staining of ZO1, which is a major component of the epithelial barrier in the large intestine. We then thought, okay, if this is associated with obesity, we can probably prevent this from happening by preventing obesity. So what we did to address this is a paired feeding experiment where we have this very obese DBDB mice, which are the blue ones here. You can see that they're gaining weight over time. And then we take their DBDB litter mates, and instead of giving them food ad libitum, we give them only as much food as the wild-type litter mates would eat. And you can see that by doing so, we prevent obesity from happening. And we hypothesized that this will also prevent infection. But surprisingly, the red group, which is the lean DBDB group, and the blue group, which is the obese DBDB group, they are basically behaving the same in terms of infection. You can see here that they are equally susceptible to mortality after Citrobacter rodentium infection. So this was surprising until we realized that these mice are also hyperglycemic. So here we're looking at glucose levels, and you can see that, of course, the DBDB mice are massively hyperglycemic, as we would expect. But these lean DBDB mice, even though their weight is the same as the black group, they're as hyperglycemic as their obese litter mates, which led us to hypothesize that maybe it's not obesity, but it's actually hyperglycemia, which is driving this susceptibility to infection. So this is exactly what we addressed in the next experiment, where we used a different model of hyperglycemia, which is injection of STZ or streptozotocin. which kills pancreatic beta cells and thereby renders them hyperglycemic. So you can see here that we achieve by STZ injection, we achieve very high levels of glucose, even though the mouse remains lean. So this is basically a type 1 diabetes model where the mouse is lean but very hyperglycemic. And now, interestingly, this is enough to make them as susceptible to the infection as the DBDB mice, because now you can see spread of this bacteria to the spleen and the liver. You can see abrogation of barrier function. Here we're staining with ecathirin. Again, you can see that these STZ mice are, have a much weaker intestinal barrier. And this is also confirmed with yet another way of making a mouse hyperglycemic by using a genetic mutation in the insulin gene. And this mouse is called the Akita mouse. You can see that these are very hyperglycemic and they're also very susceptible to spread of this luminescent Citrobacter rodentium to systemic sites. So all of these experiments, they basically mean that hyperglycemia promotes the breakdown of the intestinal barrier, and this makes them so susceptible to this intestinal infection. Even though clearance in the intestine is completely unaffected, but the systemic spread is what makes them so sick. We can also basically revert this condition or treat this condition by treating hyperglycemia. So here we're using insulin treatment, and you can see that if we treat these streptozotocin treated mice with insulin, we can normalize their blood glucose levels. And this also normalizes, for the most part, their intestinal barrier. So you can see that we can, between the blue group, which is very hyperglycemic, and the red group, which is maybe mildly hyperglycemic, we can almost fully normalize ecathirin intensity in the GI tract. So now the question is, what does glucose do to intestinal epithelial cells to so dramatically affect barrier function? So here we looked by metabolomics at various stages of glucose metabolism in intestinal epithelial cells of these streptozotocin-treated mice. So you can see that they actually have higher levels of glucose. So they retain glucose or take up glucose from the systemic circulation. And then this basically drives through the entire pathway of glycolysis all the way up to higher lactate levels. So we thought we can do a simple experiment to see if glucose metabolism is involved in driving this phenotype by blocking glucose metabolism using 2-deoxyglucose. So we basically treat these streptozotocin-treated hyperglycemic mice with 2-deoxyglucose, and just like in the case of insulin, we can prevent this massive spread of the pathogen to various systemic sites. Here we're looking at the spleen in both streptozotocin-treated mice as well as DBDB mice. So basically by inhibiting glycolysis, we can inhibit these problems from happening. So now the question is, what is the mechanism by which epithelial cells become so susceptible to this toxic effect of glucose? And when we think about glucose metabolism in intestinal epithelial cells, there are two main transporters that are relevant here. One is on the apical site, we have this sodium glucose symporter, which brings in glucose from the intestinal lumen. And then we have another glucose transporter here, which is called GLUT2, which sits in the basolateral site and is basically just passively moving glucose along the concentration gradient. So under normal conditions, glucose will be higher in the intestinal epithelial cell as compared to the interstitial site here. And then you can see that GLUT2 is basically diffusing glucose along this concentration gradient. And we hypothesized that in the case of hyperglycemia, this might actually be the other way around. There is a much higher glucose load on this side, and this brings back glucose along GLUT2 into intestinal epithelial cells. So we hypothesized that inhibiting this transport through GLUT2 might actually prevent these problems from happening. So we teamed up with a group in Paris that had generated mice specifically lacking GLUT2 in intestinal epithelial cells, and we performed the same experiment again. So we took these epithelial-specific GLUT2 knockout mice and their leadermate controls. We treat them with streptozotocin, and you can see that very similar to 2-deoxyglucose treatment or insulin treatment, we can partially restore barrier function, both in the case of ZO1 and in the case of ecathirin, by rendering these epithelial cells insufficient for GLUT2. So we can basically block this glucose influx from happening by blocking GLUT2, and we can block the detrimental side effects in the case of barrier function as well as systemic spread of the pathogen. So here again, we're doing Citrobacter rodentium infection in these mice. We're looking at the spleen and liver. Again, the streptozotocin treated control mice are very susceptible, but by inhibiting GLUT2, by genetically deleting GLUT2, we can bring the levels of the pathogen down, almost all the way down to the control level. So this brings me to the summary. We basically found that this pathway of glucose metabolism of intestinal epithelial cells, which is provoked by hyperglycemia through a diabetic state, leads to a reprogramming of intestinal epithelial cells. I didn't have time to show you all the transcriptional and epigenetic data, but basically what happens here is that there is a transcriptional reprogramming provoked by altered glucose metabolism, which then compromises this barrier function. And once the barrier is compromised, we have basically a failure to locally contain this pathogen, which then becomes systemic and spreads to different tissues. I've also shown you that we can inhibit with this, interfere with this pathway by inhibiting multiple stages here. We can either treat hyperglycemia with insulin and prevent the problems from happening. We can genetically inactivate GLUT2, specifically in intestinal epithelial cells, with the same result. And with 2-deoxyglucose, as I've shown you, we can inhibit glucose metabolism, which again prevents this loss of barrier function. So now the main question is, because I'm showing you a healthy and diabetic human here in the background, even though all the data I've shown you so far was on mice. So now the question is, do we have a similar phenomenon in humans, where would we see a susceptibility of hyperglycemic or diabetic individuals to systemic spread of these microorganisms? So to start and understand this, we recruited a cohort of volunteers. We measured all these different serum parameters in blood samples that they donated. And then what we did is we measured in their serum samples, we measured the abundance of microbial ligands by using a very sensitive reporter cell line for multiple different toll-like receptors and other innate immune receptors, which allows us to quantify the amount of microbial molecules in their systemic circulation. And then what we did here on the left side is to correlate the amount of microbial ligands that we find in their circulation with any of these other serum parameters that we measured in their blood. And what stood out here is that the #1 factor correlating with the abundance of microbial molecules is HbA1c, which is indicative of long-term glucose levels in the blood, because these are glycated hemoglobin levels. So this means that long-term glycemic control seems to be a factor contributing here to the amount of microbial ligands we can detect in the blood of these people. And you can see here one example. So this is TLR4 ligands, which is probably primarily LPS, with the levels of HbA1c, and you can see that there is a correlation between the two. What's interesting here is that very similar to the situation that I've shown you in these obese mice, BMI is actually not correlated at all with the microbial ligands. You can see here again the example for TLR4, no correlation whatsoever with BMI, and BMI is here one of the factors that absolutely doesn't contribute or doesn't correlate with the amount of circulating microbial ligands, which again reinforces our conclusion that it seems like it's not obesity by itself, but it's actually hyperglycemia or long-term failure of glycemic control in the case of HbA1c, which is driving this susceptibility to microbial influx and barrier dysfunction. So I think I'll finish here. I just want to one last time come back to this schematic here from David Schneider's lab, because I think it's really informative in terms of thinking about the relationship between pathogen load and physiological parameters. So in this case, we have basically systemic metabolic disease or hyperglycemia specifically as a factor that brings us down the health axis here. Because even under a condition of identical pathogen loads in the GI tract, we have completely different outcomes depending on the metabolic state of the host. And then very similar to the current situation of SARS-CoV-2 infection, there might be many other such physiological parameters, which is regulating the position of the host on the y-axis here, as opposed to the x-axis, which is simply pathogen load. And I think the current pandemic and many other infectious diseases as well as non-infectious diseases will really benefit from this approach of looking at physiological parameters that restore health other than just taking care of pathogen load. So these are basically non-immune or non-classical immune factors that are promoting tissue tolerance and restoration of health. So with this, I'd like to finish. I want to acknowledge all the people that were involved in this work. First and foremost, my Ph.D. supervisor and long-term mentor, Eran Elinav at the Weizmann Institute. I mentioned Eran Segal as a computational collaborator and specifically Daphne in his lab. Many other collaborators at the Weizmann Institute, and I mentioned the group of Alexandra Grossfeld and Patricia Serrades in Paris that provided these conditional GLUT2 deficient mice, as well as my own new lab at the University of Pennsylvania, which is a fantastic team of students and postdocs that are following up on these studies and addressing many more with the same concepts. And I'd also like to thank all the funding agencies that are supporting our work. And I'd like to thank you for your attention, and thanks for the invitation to speak here. And I'm looking forward to the discussion. Thanks very much.
Thank you very much, Christoph. That was really great. And so we have, as always, we have the possibility. You have the possibility to ask questions through the chat, and Jerry, we will monitor whether we see some questions. So do we have questions for Christoph Thaiss? But maybe to start with, Christoph, I mean, really all the experiments and the impact of hyperglycemia versus obesity, and obviously also in the context of COVID-19, this is extremely relevant. So, I mean, there has been a recent paper on the direct impact of COVID-19 on diabetes, if I understand correctly. I mean, on the fact that direct effect on the virus, also on pancreatic cells, and how it might. So it's a kind of a circular, vicious circle. I mean, where the virus can directly impact on the, in a way, act as a streptozotocin, if I may make the comparison, and then generate the hyperglycemia, which in turn generates the susceptibility to the infection. And so this is also obviously something that we can see with other viruses. I mean, this effect on the pancreas. So how do you see the present? It's a general question, but I mean, so we have this interplay between SARS-CoV-2 and diabetes. And do we have other interplay, other interface between SARS-CoV-2 and the intestinal microbiota? I mean, do you see other mechanisms whereby the intestinal microbiota could influence SARS-CoV-2 infection?
Yeah, that's a really interesting point. What's interesting there is that GI infections of SARS-CoV-2 were actually among the very first extrapulmonary sites in which infection was detected. I remember back probably in January or February were the first reports from China saying that they found individuals that had not only GI symptoms accompanying the infection, but also real infection in intestinal enterocytes. So in that sense, it brings up two very interesting questions. One of them is, first of all, what does GI infection contribute to the overall outcome of COVID-19, which I don't think is fully resolved at the moment, based on the literature. And second of all, as you point out, is what would be the contribution of the microbiome to not only the susceptibility to enteric infection, but also the systemic outcome. From our perspective, of course, it's very interesting what the microbiome is doing in terms of metabolic modulation of the host, because there is such strong data that the metabolic status of the host is a main driver of susceptibility. But for the other part, which is what the microbiome might do to regulate enteric infection of SARS-CoV-2, I would tell you to stay tuned for Mayan's talk in a couple of weeks, because this is something where her group had quite interesting discoveries recently.
Okay, we will wait. Thank you. Do we have some questions coming?
That was a real tour de force, and I am an obesity expert in the world, and believe me, I've been in it for 40 years, and you covered so many different models that I won't even tackle them, but I look forward to talking with you a lot more.
I'd be happy to.
Yeah, it was really exciting. I think the diabetes COVID connection it may be the weakest of what you presented. So, and I'll say why. Most people with diabetes are not dying when they get COVID. It's the ones that are old, have had it severely, are insulin treated, and literally have every complication in the books. Heart problems, vascular problems, everything. So be real careful about that connection. I'm just cautioning you. I think COVID is uniquely attacking frail people, meaning killing them, of every frailty. So anyhow, for what that's worth, I would make one other comment. As you start manipulating the nutrient composition, and I paid especially big attention to your Biggest Losers, there's no question from my conversations with Jeff 10 years ago that the composition of the diet makes a huge difference. And with the rodents and the high-fat diet, you have an extreme diet. I want to emphasize that. It's a terrible model for humans. They do never have that kind of amount of fat. And if I were a mouse or a rat taking that amount of fat, I'd be sick as a dog. So I would caution you on that particular extrapolation. I think the composition is extremely important for the microbiome, even small differences in composition. And so it's a little hard to predict, exactly how that interaction is taking place. But it's a very unique model with many, not just a few, many differences from human obesity. So I'll get to my question. I think these studies really should be expanded right now to humans, because we have humans who are obese. We have humans with diabetes at every stage, mild, middle, moderate, on insulin, not on insulin. And I think the human model of humans is by far the best.
Yeah, absolutely. Let me briefly comment on all 3 points that you made, because all of them are excellent in their own right. So let's start with diabetes and SARS-CoV-2. Of course, all the data I've shown you was not in the context of SARS-CoV-2. All of it was in the context of diabetes, hyperglycemia, manipulation of the intestinal barrier. Whether this is relevant for SARS-CoV-2, we don't know at this stage. But nonetheless, I think it's interesting that if there is one factor that was unanimously discovered across all epidemiological studies so far, in terms of what predisposes to severe COVID-19, it was always diabetes and hyperglycemia. There's not 100% agreement on hypertension. There is not 100% agreement on neoplastic diseases, cancers, and so on. But diabetes always comes up as one of the strongest risk factors, in addition to old age, of course. So I agree with you
on that, because 30% of people over the age of 75 have diabetes. So you have to be real careful about that confounding. It's right. Confounded. Old and diabetic are the two worst things to be and get COVID.
I agree, exactly. But let's go to the second point, which is another really good one about dietary composition. So the point is really well taken that and this is something we've seen in our own hands, that minor changes of macronutrients or even micronutrients alter the composition of the microbiome. The reason why we use the classical 60% high-fat diet in this case is because this is what Jeff had done his original studies with, and what many people use to generate obesity. I should say that we have done, in addition to the data that I've shown you today, we have done more controlled experiments where we basically used custom-made diets that are that are macronutrient matched and only vary in the fat content, protein content, or carbohydrate content. And this effect of basically longevity of the diet-induced microbiome changes, they are seen across all diets. So basically, when you have an initial dietary perturbation, in this case, it's associated with obesity, but it's not necessary, it doesn't have to be the case. And we basically cease this dietary intervention and we let the metabolic parameters go back to normal. The microbiome normalization will lag behind, regardless of the dietary composition that we do. And this is in line with some of the early work from Peter Turnbow, where he showed that very rapid dietary changes, not in a chronic model like we did, but very rapid dietary changes also provoke this phenomenon that the microbiome doesn't catch up with these repeated cycles of dietary changes. And then the last point you brought up about humans, I couldn't agree more that this is exactly what we have to do. We have to study microbiome recovery and the role of the microbiome in weight loss programs, basically clinically guided weight loss programs, to see whether we can, similar to the situation I've shown you for mice, if we can use microbiome information to predict the long-term weight maintenance. But if we can predict, can we use microbiome information to do something about it? So that's, I'm very impatiently waiting to start these kind of studies on basically humans undergoing these weight gain, weight loss cycles.
I hope you'll be here to do those studies.
Mm-hmm.
I hope so too.
Okay, thank you very much. I believe that we are coming actually to the end of the webinar timing. Do we have a final question for Christoph? Apparently not. Okay, well, thank you so much, Christoph. That was really great. And
thank you.
As Barbara has said, we really hope that when COVID-19 will slow down, which is not the case presently at all now. Especially in Florida. Especially in Florida, but it will come someday. We can welcome you and also with Mayan Levy. So thank you. That would be great. Thank you all for attending this seminar, and we keep in touch and be safe, as we keep telling you. Bye-bye. Thank you.
Thank you again. That was a great lecture.
Thank you.